Hey guys, in this video we are going to study about the digestion and absorption of proteins. First, moving on to the mouth area. So the juice present here is saliva, whereas there is no proteolytic enzyme present in the mouth. That is, there is no presence of proteins in the mouth. So there is no proteolytic enzymes. There is presence of no proteolytic enzymes. Whereas moving on to the substrate, so the substrate is same as that of the carbohydrates, that it converts the polysaccharides into disaccharides, that is dextrin and maltose. Moving on to the second one, that is the stomach. So in the stomach there is presence of the gastric juice. So second one is the stomach. In stomach there is gastric juice, which has the enzyme pepsin. So pepsin is the one which converts the proteins into three end products, that is proteoses, peptones as well as polypeptides. So in the stomach the gastric juice has some enzyme called as pepsin. So the pepsin is the one which converts the proteins into proteoses, peptones, 3Ps, as well as the large polypeptide. Remember it as 3P. Further moving on to the small intestine, same. The small intestine has main two juices, that is pancreatic juice as well as the succus entericus. So third one is the small intestine. The small intestine mainly has the pancreatic juice. So the pancreatic juice is the one which has three enzymes, that is trypsin, chymotrypsin as well as carboxypeptidase A and B. First one is trypsin. Second one is chymotrypsin. And third one is carboxypeptidase A and B. So the trypsin and chymotrypsin has same substrate. It has the same substrate, that is the end product of the gastric juice, that is peptones as well as proteoses. It includes the peptones as well as proteoses. The end product is easier one, dipeptide, tripeptide as well as polypeptide. It is dipeptide, tripeptide as well as the polypeptide. Moving on to the substrates of carboxypeptides A and B, that is the end product of the pancreatic juice. It's a easier one. For trypsin and chymotrypsin, it is the end product. The substrates are the end product of the stomach, that is gastric juice. For carboxypeptidases A and B, the substrate is the end product of trypsin as well as chymotrypsin, that is dipeptides, tripeptides and polypeptide. And the end product is the easier one, that is amino acids. Yeah. So further moving on to the succus entericus. So we'll make a chart so that we can understand it in a better way. So further moving on to succus entericus part. So the enzyme mainly involved is dipeptidase, tripeptides, tripeptidase and aminopeptidase. First one is dipeptidase. The second one is tripeptidase and the final one is aminopeptidase. So these are the one which converts into dipeptide, same tripeptide as well as polypeptides, and the final end product is amino acid. It converts into dipeptides, tripeptides as well as large polypeptide, and the end product includes the amino acids. This is the diagrammatic representation of the digestion of proteins. Now we'll move on the detailed explanation of digestion of proteins. So in the digestion of proteins, first remember one thing. In the digestion of carbohydrates, the enzyme for the digestion of carbohydrates was called as amylolytic enzyme. So in the digestion of proteins, there is an enzyme involved which is called as proteolytic enzyme. So what is proteolytic enzyme? So proteolytic enzymes are the one which helps for the digestion of proteins. It helps for digestion of proteins. First we'll look after the digestion in mouth. Digestion of proteins does not start in mouth because the saliva does not contain any proteolytic enzyme. So the digestion of protein mainly starts in the stomach. So this is an important point. So digestion of proteins, it does not start in mouth. Why? Because saliva has no proteolytic enzymes. Therefore, where does the digestion of protein starts? It starts in the in the stomach. Because the the gastric juice has proteolytic enzyme. Okay, this was about the digestion in mouth. Moving on to digestion in the stomach. Studying about the digestion in stomach, pepsin is the only proteolytic enzyme in the gastric juice present. Renin is also present in the gastric juice, but it is absent in human, but it is found in baby young ones, which is used to feed. It is present in the milk. So, pepsin is the only proteolytic enzyme present in gastric juice. Renin is absent in humans. It is not present in humans, but it is found during pregnancy for feeding infant. Next, moving on to digestion in small intestine. Looking into digestion in small intestine. Most of the proteins are digested in duodenum as well as jejunum by the help of proteolytic enzymes. There are two main juices, as I have said, that is pancreatic juice as well as succus entericus. We'll see in both of the juices. We have to remember this point. Most of the proteins, where are they digested? They are mainly digested in the duodenum and jejunum with the help of proteolytic enzyme, and there are two main juices. We'll write about it now. So in that, the first one is pancreatic juice. Pancreatic juice, as we have spoken, it contains three main enzymes, that is trypsin, chymotrypsin as well as carboxypeptidases. In these three enzymes, the trypsin as well as chymotrypsin are also called as endopeptidases. What is endopeptidases? So these are the molecule peptide bonds. So it breaks between the molecule. So it thus it is called as endopeptidases, whereas the bonds are broken between the molecule. So the pancreatic juice consists of the trypsin, chymotrypsin as well as carboxypeptidase A and B. The trypsin and chymotrypsin is also called as endopeptidases. It is also called as endopeptidases. What are endopeptidases? Breaks the peptide bond within the molecule. It breaks the peptide bond within the molecule. Next, moving on to the succus entericus. Succus entericus. The enzyme present here is easier one, that is dipeptidase. Tripeptidase and aminopeptidase. So this is about the digestion of proteins, which is usually asked for 5 marks, sub question in the 5 marks session. Further, moving on to the final products of protein digestion. Final products of protein digestions are amino acids, which is absorbed into the blood from intestine. Further, moving on to absorption of proteins. So looking on to the final products of digestion, so final product of digestion of proteins, that includes the amino acids, so which is absorbed from the small intestine to blood. So looking into the absorption of proteins, so the absorption of proteins is mainly by the amino acids from the small intestine. It is absorbed in the form of amino acids from small intestine. So there are two types of rotational amino acids. So in the amino acids, there are two types of rotational amino acids, that is levorotatory amino acids as well as dextrorotatory amino acids. So it concludes the levorotatory amino acids as well as the dextrorotatory amino acids. The levorotatory amino acids are actively reabsorbed by the means of sodium cotransport. So it is reabsorbed by sodium cotransport, and dextrorotatory amino acids are absorbed by the facilitated diffusion. So another important point is absorption of amino acids is faster in duodenum and it is slower in jejunum and ileum. So that's it about the digestion and absorption of protein, where they'll be asking the combined question, that is digestion and absorption of proteins, for 5 marks. So this is a quite important question. So hope you have understood this video. So please do like, comment, share and subscribe. Thank you.